Thank you for joining us for the webinar series by IIT Alumni Center, Bangalore. So the center provides basically a unique networking platform for both the academia industries, and also it bridges the gap between the two or among the academia or industries through the projects of national importance. So in this webinar series, we have a lot of interesting topics, and the today's webinar is on discussing electrochemical wearable sensors. So not just for biomedical application, but also we are going to hear about the areas like agriculture and security. So that's pretty interesting. So the today's speaker is Dr. Yugender Goud Kotagiri. So he received his MSc and PhD degrees in chemistry from the Department of NIT Warangal. And he also worked as a postdoctoral researcher at the UCSD, University of California, San Diego, and also Johns Hopkins University in US. So this was during his years of 2018 to 2022. And currently he is working as a Ramanujan Fellow at the Department of Chemistry, IIT Palakkad. So he has a very successful career so far. He has published 55 journal papers in both international and national journals. And his name was featured in the world's top 2% scientists database by Stanford University. And this was for the year, 2 years actually in continuation, 2021 and 2022, in the subject category of analytical chemistry and energy. So very impressive, congratulations. And this talk will be moderated by Dr. Koteshwar Reddy, who is currently working as a research scientist in the Identify Sensors Biologics Corp USA. And so it's very early morning for him. And he got a PhD degree in chemistry for the research work on biopolymer stabilized metal nanoparticles towards the development of electrochemical biosensors, again from NIT Warangal. And his current research interests are quite wide, but all focusing towards the electrochemical biosensors, energy, corrosion, and biomedical applications. So this webinar will be co-hosted by Dr. Sushila Venkatraman and myself. And for audience, very important, please post your questions in Q&A box. So that will be taken up during the discussion, during the short breaks, during the talk itself. So over to Yugender.
Thank you, Professor Abba, for the introduction. Sharing my slides. So the presentation is visible, right?
Yes.
Professor Abba? Okay.
Yeah.
So thank you, Professor Abba, for the nice introduction. So today I would like to talk about the wearable electrochemical sensors for healthcare, agriculture, and security applications. So whenever we heard about the wearable sensors, so in our mind, one picture that came into that. What is the smart wearable watches and wristbands, smart wristbands. So, but those we are calling as a first generation of wearable sensors, and those mechanisms mostly based on the physical and biophysical based sensor mechanisms. So, but in today talk, I'm going to talk, majorly focusing on the wearable electrochemical sensors. These we are calling as a second generation electrochemical based sensors. In the first generation, wearable sensors, majorly we are focusing on the detection of the temperature, stress biomarkers, oxygen levels, heart rate, various things. But here in electrochemical wearable sensors, majorly we are focusing on the continuous monitoring of the biomarkers that are related to the chronic diseases such as diabetes, cancer, renal, etc. And one more thing is that we are also focusing on the continuous monitoring of the drugs that we're calling as a therapeutic drug monitoring. And these are mostly related to the biomedical application and healthcare applications. But in the wearable electrochemical sensors, not only this biomedical, it is also useful for the agricultural applications and security applications. The agricultural application means you can continuously monitor the moisture, water level, pH level, potash, carbonate levels in the soil or near the paddy fields or on top of the plants and leaves. That is related to the agriculture, wearable agricultural electrochemical sensors. And in the security applications, that is also related to the mostly for detection of the nerve agents or toxic molecules on textile-based sensors. That is also one wearable electrochemical sensor type. So in today's in webinar, in this talk, my talk is continuous in this flow. So initially, I would try to explain about the introduction of the biosensor and what are the basic components of the biosensors. And later, I will enter into the electrochemical sensors. What are the electrochemical sensors and the types? And later, I will enter into what is the importance of the wearable sensors, why wearable sensors came into the pictures in the electrochemical field. And I will also explain about the various sensing platforms available in the wearable electrochemical sensors and how we can apply those to the various type of the fields. I will explain. And later, I will try to explain a little deep about the microneedle based sensors in biomedical applications. Microneedle sensing technology is one of the major technology that can useful for the continuous monitoring of the various drugs and also the drug delivery. And later part, I will try to explain initially little introduce about the tattoo-based sensors and textile-based sensors. And along with that, I will also try to explain about what are the future possibilities of these wearable electrical sensors, especially in these three fields. So coming to the biosensors definition and the basic component. So sensor means a sensor is a device so that can specifically detect these molecule that we are calling as a sensor, right? So the biosensor, so with the help of the biorecognition elements, those are enzymes or antibodies or aptamers, whatever the biological components. So with the help of that, we are specifically detecting the chemical and biological compounds. Okay, and these interactions we are monitoring with the electrical and thermal and optical signals. So all these combining the three systems we are calling as a biosensing systems and biosensors. So when you see the right side diagrams, whenever we hear about the biosensors, electrochemical sensors, the first thing in our mind, it will come to the picture the glucose sensors, glucometer. So here the glucometer we can see. So the glucometer, what is one small device is there and one sensor strip is there. So when you pick your plate and cast on the top of the electrode surface, so within the minutes you will get the output of your glucose readings into the instruments. So these devices can basically work on the electrochemical amperametric sensors and enzymatic sensors. And one more biosensor is that we know that the pregnancy test kits that is based upon the antibody ELISA-based technology. So these are the point-of-care sensing techniques. Means every time we need to take this sample, either blood or within external biofluid samples, and we need to add on the top of the electrosurfaces, and with that we can get the values. But here the wearable sensors means those will continuously monitor the analytes inside the biofluids, the body biofluids. So one is the best example, the commercial device available here, continuous glucose monitoring devices. So here, imagine you have a device, so you are wearing into the body, and this device is continuously monitoring your glucose levels, either in the sweat or interstitial fluid, and you are getting the readings to your mobile phone. It will act as a personal device to you. So based upon that, you can take the intake, whatever the insulin intake you want to take it, or tablets you want to take it, or you want to go to the hospital for doctor checkup, you can take it. So these are the major thing in wearable sensors. It's continuously monitor the unlines. So before going to the deep into the wearable sensors, one more time I will explain the components of the biosensor here. So biosensor has the three basic components, right? One is the target analyte. So what we are going to detect that we are calling as the target analyte, and the recognition elements. So those are the chemical or biorecognition elements. And one more is the physiochemical transducer. So that can detect the signals between the target analytes and the recognition element. So target analytes means, so what you're targeting. So that we are calling as target analytes. So it is a biomedical means we're majorly focusing on the biomolecules detections, biomarkers, neurotransmitter detections. This will be majorly helpful for the chronic disease management. And the other target analytes, pharmaceutical drugs for therapeutic drug monitoring, and forensic drugs, illicit drugs, and food analysis, mycotoxins, pesticides, herbicides, environmental pollutants, toxins, volatile organic compounds. These are the different target analytes. Those we can detect by using the electrochemical sensors. And the recognition elements, as I said, the chemical and biological recognition are available. So biological recognition elements, enzymes, antibodies, lipids, cell organelles, and aptamers. Okay, and these synthetic recognition elements, molecular integrated polymers, and conductive polymers. So apart from these, we have nanomaterials also, 2D nanomaterials, hybrid nanomaterials, functional nanomaterials, that also will act as a recognition elements. So the interaction of these target analytes and the recognition elements, we want to measure with the some techniques we needed. So those techniques is transition techniques we're calling. So the transition techniques either electrochemical or optical or mass-based techniques. But this presentation is mainly focusing on the electrochemical based techniques. So coming to the electrochemical sensors principle. So electrochemical sensors are majorly three types. One is the voltammetric sensors, potentiometric sensors, and ampermetric sensors. So in the voltammetric sensor, so what we have here, we have an electrode and electrolytic solution we have. So in electrolytic solution, electroactive molecule we have. So those electroactive molecule can possibly to undergo oxidation reduction. Okay, so whenever we apply the potential energy from the instrument, the potential stat to the electrode surface, so the electrode energy levels are varying while comparing to the electroactive compounds. So because of that energy level difference, the electroactive molecule can flow near to the electrode surface and undergo the redox reactions, whether oxidation or reduction reactions. And while oxidation reduction, the electrons are releasing, those electrons we are measuring as a current value from the output of the voltammetry techniques. So this voltammetry, either the cyclic voltammetry technique or pulse voltammetry techniques, differential pulse voltammetry technique, or square wave voltammetry techniques. So while increasing the concentration of the analytes, so obviously we'll get the increase in the current values. By using this principle, we can develop the voltammetric sensors for variable biomarkers and form scriptlets. So the second kind of the sensor, the potentiometric sensor. The potentiometric sensor, majorly we are using the ion selective membrane we are using. So those are organometallic based compounds. So those four sides have the possibility to attract the specific kind of ions. So we have commercial ion selective membranes are available for the H plus, potassium, chlorine, fluorine. So those ion selective membrane you can put on the electrode surface, and by using the reference electrode, with the help of that potential change, you can measure here. Means whenever it attract the ions, whenever it is entered into the cavities, it depends upon the concentration. The potential gradients also will vary. So that potential gradient variance we can measure with the potentiometric techniques. So while increasing the concentration levels of the ions, we can get the fluctuations in the potentials. Those fluctuations will correspond to the concentration of the ions. By this way, we can measure the ions. These sensors we are calling as a potentiometric sensors. One more is the amperometric sensors. These are also similar to the voltammetric sensor, but here here we are the scanning is of the potential is from one range to another range. But here in amperometric sensors, basically initially we can try with the voltammetry and we can find out in which potential range we are getting the targeted light peaks. So then we can take the potential of that potential and we can apply in the amperometric technique here. And amperometric techniques are not only the chemical based techniques. We can use the enzymatic, aptamer, and antibodies techniques. Suppose the example here is the glucose sensor example. So we have glucose oxidase that is coated on the electrode surface. So in presence of the glucose oxidase, the glucose is converted to the gluconic acid and releasing the peroxide ions in presence of the oxygen. So those hydrogen peroxide is the attractive molecule that can reach the electrode surface and it is undergoing the redox reactions. So those redox reactions, electron behavior, we are measuring with the amperometric principle. This we are calling as a amperometric sensor. So coming to an ideal electrochemical biosensor should have all these characteristics. Means it should work in real-time conditions and easy of operations, and it should be affordable and should be more specific to the target analytes. And moreover, it should be user-friendly. Means any bare person can easily use. And one more is a rapid and robust and medium-stress instrumentation. If you have all these characteristics, we are calling as these are ideal characteristics, and your sensor is a reassured sensors. But to achieve all these things, we have some challenges are there to achieve this one. Okay, the main thing is that a real sample matrix analysis. So whenever we develop the sensors in the laboratory, we are doing in the buffer conditions. Means we are taking the simple buffer and some electroactive components we are adding that, and some supporting electrolytes we are adding that. So in that conditions, we are getting our sensor is getting the good repeatable readings, good readings. And but when it coming to the real matrices, the blood or urine, any other biofluid matrices, so in that case, so in the blood where the bulky structures are there, heavy protein molecules are there. So in presence of the proteins, detection of our undiet is another target thing. Okay. So that we can overcome with the by use of the different kind of the functional polymer layers under the electrode surface, or either by using the microfluidics nanofluidics technology, we can try to avoid the bulk protein molecule initially, and we can only allow the simple matrices on top of the electrode surface. By that way, we can avoid. And one more thing is the biofouling studies also. Means initially your sensor will give the good response, but when you want to measure for a lot of times, the sensor will response will be decreases. Means the bioregression elements can fall from the electrode surface. So to reduce that one, we want to use some anti-fouling coating layers. Those are the polyethylene glycol, zootronic layers, or agrogel, different type of gels we can use. And one more challenge is sensitivity and selectivity. So these can be achieved with sensitivity by combination of the integration of the hybrid nanomaterials with the bioregression elements. We can achieve the good sensitivity. And these we can achieve with the for the point of sensors. But one more challenge is the continuous monitoring of the biomarkers and therapeutic drugs is the one more challenge, because if you have all these characteristics also, but you are measuring the your biomarker concentration, or you are measuring your drug concentration at one time, but you want to measure the continuously. So for continuous measurements, the main thing is that the availability of the biofluid is important, because when you're taking the blood and adding on the top that you are measuring, but after that you cannot use the same blood for so much time. Again, you want to take the blood from the patient, the whatever you want to test, or you want to test. So for that, the continuous source of the biofluid is very important. So in that case, the wearable sensing technologies came into the picture. Okay. So here in the wearable on-body sensors, so the majorly is focusing on the not the conventional biofluids. The conventional biofluids majorly focus on the blood and urine. Means those are we are calling as a external biofluids. We are taking and we are measuring. But the wearable sensors may focusing on the interstitial fluid. So interstitial fluids under the skin. It's every time it's available. If you access the interstitial fluid, so you can continuously monitor the biomarkers levels and drug levels inside the body. And sweat is also one of the good source. You can measure the various kind of biomarkers and the unlights and tears and saliva. So these are the different kind of sensing platforms: tears, saliva, ISF, and sweat sensing platforms. So as we know, this is one of the trending research topic. It's emerging research topic. So in 2019, 2020, this the market value in USD is 32 billion, but it's going increasing very rapidly. It's going to reach by 120 billions by 2029. So these are the various wearable sensing platforms can possible to work on that. So these are not in the developing state. Some of the devices are available in the market also. So these are the listed devices here. So those are focusing on the ISF monitoring of the glucose, blood glucose, glucose in ISF, and sweat. So these are the commercial devices available in the market. So these are not in the developing stage. Some of the devices are also available. So coming to the wearable various techniques. So what are the types of wearable sensing techniques are available? Here means these are the 4 or 5 different types of sensing techniques are available. So those are microneedle, tattoo, textile, microcatheter, glove, these sensors. So coming to the microneedle. So the microneedles are tiny, sharp, and kind of needle devices. So typically the size is in micrometer. So it can easily pierce into the skin in minimal invasive way and non-invasive way, and it can access the analytes under the skin. So in that way, we can detect the analytes, and even we can continuously monitor the analytes. Okay, those are the, so it can majorly useful for the screening of the various type of analytes in ISF. So those analytes should be the therapeutic drug monitoring detection. And this microneedle sensing technology not only for the sensing, it is also useful for the drug delivery. Suppose imagine you have a several kind of array of needles are there, and some needles are detecting the biomarkers levels. In other needles, you can inject the drug. What are the respective drug is needed for the patients. So by this technology, we can detect and deliver the drug also. And one more is the tattoo-based sensors, the wearable sensor. So the tattoo means you have some adhesive paper, you can take it, uh, and you can print the sensor on the top of that, and you can wear under the skin. Okay, and these electrodes you can connect with the flexible potential stats, and by this way, then you can generate the sweat. You can on the top of the body by using the exercise or some methods also there to produce the sweat from the body. So by that way, you can measure the unlights. You can screen the unlights in the sweat, and one more, the major advantage of the status sensors is that not only for the sensing, so we can by using the sweat source, you can produce energy also, because sweat is also one of the good abundant source of the biocomponents. Lactate is there, glucose is there, fructose is there, several biocomponents are there. So by using the bioionides and biocathode enzymes, you can cleave those molecules, and by cleaving that, you can produce energy. So there's a one more trend of the research is there, self-powered wearable electrochemical sensors, so that can helpful for produce energy to the wearable devices. And one more wearable sensing platform is the textile wearable sensing platform. Means here, so you can print the sensors of the textile, the body, and imagine those sensor has equipped with the one biosensing assay, either bio or chemical assay, and that can specifically detect the target analytes. Those are the toxic gases or vapor gases. So you have an industry. So in industries, anytime if any effluent toxic gas are releasing, so the worker has the sensors on the body. So whenever it reaches on the top of the electrode surface, it can give an alert to the workers, the factory workers. In that way, it will be useful in the industrial purpose. And one more thing is it is also useful in the security purpose. So nowadays, the terrorist organizations, they're using the bio warfare agents as a chemical weapons. So nerve agents they're using as a chemical weapons. So the nerve agents you can make a sensor assay on top of the textile, so that can cleave the nerve agents, and whatever the byproducts those released by that after cleavage, those byproducts you can detect by the electrochemical techniques, and you can get the initial alert of the nerve agents exposure. So that is also can be useful for the security applications. And one more is that microcatheter is also one of the good platform. So you can microcatheter is the tiny tubes. It is in the micrometer to millimeter range. So these are useful mostly in the ICU conditions with the doctors. So they can inject the various type of IV fluids and the liquid drugs also they are injecting in that. So imagine you have a sensor along with that microcatheter, and that can monitor your drugs on real-time condition in ICU conditions. So one good thing is that uh, anesthesia drugs, the propofol, fentanyl, mostly they are using for ICU conditions. But if you use the high amount of these anesthesia drugs, that can lead to the side effects to the patient. Sometimes it lead to the death also. So during the surgery time, you have a device of the microcatheters that can detect the anesthesia drugs effectively. So in that way, you can give the anesthesia drugs whatever how much you want, based upon the feedback of the microcatheter sensor. This is also one possibility in biomedical field. One more is the glove-based sensor. Here, these glove-based sensors mean the sensors you can print on the glove, and you can integrate them with the flexible potentiostat on the wrist. And those are mainly useful for the detection of the powder samples. Means, so nowadays, like illicit drug transport is also one of the major thing in India, right? So whenever you find any unsuspicious compound, illicit drug compound, always we are depending on the centralized instrumentations. We are sending a sample to them. So imagine you have a sensor, the glove-based sensor. When you touch the sensor with the powder, at least if you getting the initial reading of the what are the components are there, what is a morphine or cocaine, methamphetamine, fentanyl, what are the components are there, if you get an idea. So that is also very helpful for the especially for security applications. So these are the different platforms I explained, but now I'm going to explain little thoroughly about the microneedle sensing platforms. So as I said, microneedle sensing platform we are calling as a lab under the skin. why we are calling as a lab under the skin means, so whatever the unlights we are detecting in the laboratory, whatever the unlights we are detecting in the laboratory condition, external conditions, those are all the type of unlights can possible to detect in the, under the skin. So that we are calling as a lab under the skin. So typically these microneedles are in micrometer range, in 20 to 50 to 100 to 1000 micrometer range. So those can easily pierce into the, and one more thing is that here, these are the minimal invasive, okay, and non-invasive. These are not the pain. How it is minimal invasive means, you can see here. So the traditional needle, whenever you insert into the skin, it will go onto the dermis part. So that's why you will get the pain. But whatever the needle microneedle we are talking about, that will go into the stratum part only. So you will not feel that much pain here. So that way we are calling as a minimal invasive technologies we are calling. And one more thing is that what are the unlights possibilities are there in the blood? Almost 95% unlights are present in the ISF, in interstitial fluid. Only the concentration varies will be different. Maybe in the blood you will found 10 times, maybe you'll find in the ISF in the 5 times. And some type of unlights or even accessibility is more in the ISF compared to the blood. So the ISF is also one of the good source to use as a biofluid to monitor the various type of the biomarkers. So in this, I'm going to explain on the recent paper. It is published in Nature Biomedical Technology. So here, this is an integrated wearable sensor to monitor the multiple biomarkers in interstitial fluid. So here you can see, so they develop the microneedles and they develop their own potentiostat integrated device and they wear it on the body. And with this work, they said that this is my previous group already. So they said that they continuously monitor the lactate, glucose, and potassium and ions also. So initially, they printed the microneedles with the micromachining method. So there is a different type methods out there. You can use any 3D printing methods, photolithography methods, any 3D printing methods you can use. So here, initially they designed the sensors with the 3D AutoCAD or Fusion 360. So then here, they printed the sensors on top of the silicon dioxide filling. So they use the biopolymer, and later they the tips of the sensor they coated with the silver, gold, chromium, and silver and gold. They coated. Okay. So here, majorly the three steps are there. One is the micro machining step, and one is the thin film deposition step, and assembly of the techniques. Means initially to make the arrays of the electrodes, and later deposition of the conductive metals, the chromium, silver, and platinum, and later the assembly of the electrodes. These are the three parts in the microneedle sensor making. And after making the sensors, so they want to modify the sensor surface with the enzymes. For glucose detection, they use the glucose oxidase as a bioregulation element. And for lactate detection, they use the lactate oxidase as a bioregulation element. And alcohol detection, for alcohol oxidase, they used. And they also use the PPT as a anti-fouling layer, polyphenol diamine coating layer initially on top of the electrode surface. So that can eliminate the possible bulky structures. I mean, it cannot attract the bulky protein structures. It can reflect those protein structures. In that way, they are eliminating the bulk proteins. Okay, and later part, the enzyme they're immobilizing, and top of the Nafion they're using. The Nafion using as an anti-fouling coating layer, so it can protect the bioregulation elements on the top of the electrode surface. It cannot easily fall from the electrode surface. So here, suppose example I'm showing is alcohol oxidase. The alcohol, whenever in presence of alcohol oxidase, it's forming the hydrogen peroxide. Those hydrogen peroxide is attractive molecules, so that we can monitor with the electrochemical amperometry technique. So initially they studied the in vitro group studied the in vitro studies, and the sensor getting the good reproducible results, and we calibrated our sensors, and we developed our own app. So that app can give the respective target and dex readings. So later we studied the on-body patients. We 5 people in our lab voluntarily we wear the sensors outside in the body. So for the lactate, what we did, like we do the exercise intensively, and we can check the lactate levels in the different body patients. So whatever here, the dotted lines here showing that, so that dotted lines we are measuring by using our developed sensors, CWS sensors. But whatever the red points that we are getting, that were the commercially available lactate sensors are there. So we correlate with that. So almost the values are correlating. In similar way, the glucose sensors also, we take the high intents of the glucose products like cool drinks and burgers we have eaten, and we check our glucose levels. So whatever our device is giving glucose levels, we compared with the commercial glucometer also, and almost the levels are matching. And the similar way, the alcohol also, with the alcohol meter we tested, and with our sensor we tested, the output readings are matching. So, but these human studies we are did in only the pi subjects, but we want to carry over more. We want to study the different age people, different gender people, different kind of region people to further develop this device. So you can see here the video, how we did here. So the right side part, so that we have as a battery and potentiostat, miniatures potentiostat, and the left side part we have a 3D printer, and we have adhesive layer. So when we remove the tape from the adhesive layer, and we can easily wear onto your arm, and you can just press it, and you can connect through your phone. Okay, so whenever you connect your phone, then you can activate what that is a bipotentiostat. So what electrode you want to focus. So then you can measure with the amplitude reading, and with respect to the current values, we can convert them into the concentration of the respective lactate, glucose, or alcohol oxidase. So in similar way, we also developed the another prototype microneedle sensor for levodopa. The levodopa is one of the gold standard drug that can be useful for treatment of the Parkinson diseases. So Parkinson disease is one of the important neurological disease. It's one type of the chronic disease. So the drug intake is varies from person to person and depends upon age also. So the nanotherapeutic addition of this drug is very important for effective management of the Parkinson disease. So for that, we developed the prototype microneedle sensor. So this is the orthogonal detection. We have a two electrode system here. One electrode is modified with the enzymatic assay, and one electrode is modified with the chemical assay. And in enzymatic assay, we detect with the amplitude technique, and non-enzymatic assay, we detect with the square wave automated technique. So in enzymatic assay, the tyrosine enzyme we cast it on the electrode surface. So those enzymes specifically create the L-dopa molecule dopaquinone, and the dopaquinone oxidation peaks we are observing in the amplimetry. And the direct way is also dopamine is catalyzed by the material, the chemical materials. Those behavior also we are measuring with the pulse oximeter techniques. Then we successfully demonstrated with the buffer conditions and ISF conditions, and we also demonstrated with the skin mimicking models and gel mimicking models. So these works I carried out during my postdoctoral stay. But whenever I joined here in IIT Palakkad, so I also focused on the neurological disease management. So I focused on the detection of the serotonin. Serotonin is one of the important neurological biomarker. So here I went on with the chemical sensing assay. So the working electrode is modified with the silver nanoparticle reduced graphene oxide chemical assay, and that is finally we are using neoprene as a anti-polyp coating layer. So whenever the serotonin oxidation, we are monitored with the square wave voltammeter technique and amplimeter technique. So then we successfully demonstrated the sensor in the buffer solutions and ISF solutions. So interestingly, our sensor but could respond in the micromolar conditions. And we also tested the performance of the continuous monitoring. So we tested the device in a particular concentration that is around 40 micromolar concentration, and we tested for 3 hours. So interestingly, the sensor is giving the almost 95% recovery current values from starting value. So that means that the sensor is responding up to the 3 hours continuously. You can measure the readings every 5 minutes or every 10 minutes. So recently, this paper we published in Basis of Nanomaterials. So maybe till now I explained about the microneedle sensors. So now I can invite Kodeshwar Reddy. So the comment on what are the challenges about the wearable sensors, what are the things we can improve, how we can commercialize this type of sensors? You can comment on Koteshwar. So Koteshwar, you're online, right?
Yeah.
Okay.
Thank you for the nice presentation. Yeah. So in continuous monitoring, we always face one serious problem, like biofouling of the recognition material on the surface. How do you address that? Can you comment on that, please?
Okay, so as I said in the initial slide, so biofouling is one of the important thing, because for initial readings, for you want to check for one reading, two readings, your bioassay can work in good way. But you want to continuously monitor the same levels for hours and hours. Sometimes you want to aim for the days. So that time the sensor should be very stick on the it should be stable on the surface, electrode surface. For that, maybe you want to use the some kind of the functional polymer materials in on the electrode surface body, so that way you can protect your biological element, whatever enzyme, whatever the aptamers, those you can protect on the surface. Okay. On the bottom you can use some kind of functional nanomaterials, and at the same time on the top also, you can use one kind of material or falling materials. So mostly negative charge materials or negative positive charge deuterium materials that people are using. So that way, in both way it can help. It can protect the layer, and one more way is that it can also help the to eliminate the molecules. Means what are the bulk molecules enter into that one, it can eliminate. So mostly the target biomarkers molecule, whatever you're focusing on the small molecules. So imagine that suppose you are using one agar gel, or maybe you are using one polyvinyl chloride type of material. So those gels have the little cavity type molecules. So those can allow only the small type of molecules inside to the electrode surface, small molecules. So whenever you have the bulk proteins like albumins, globulins, PSA proteins, it cannot enter into the that gels. So it cannot reach about the electrode surface. So in that way, the two possible. So you are eliminating the matrices of the bulk matrices. You are eliminating. At the same time, you are allowing the specific target small molecules. You are allowing that. Okay. So that we want to plan actually. So there is no ideal material. This material you can use. This material you can use. So depends upon your unlike behavior. So that by that way we can plan. So what type of anti-fouling coating layers we can use. What type of layer we can coat on the electrode surface that we can use.
My next question
Okay.
would be, for example, in buffer mediums, in artificial mediums, we will get the response. But in the reality, let's take a clinical sample or agricultural or any other samples. Those will be much complicated. How do you address that?
The same
How do
thing,
we get?
whatever I said. Okay, same. Like whatever I explained now, till now I explained about the biofouling, right? So similar things, so that will help you to the in later part of my explanation, I talk about the coating layer, anti-coating layer, right? So that is the one way to eliminate the molecules. What are the interfered molecules? That is the one way. And one more is that tag-based technology means. So in electrochemical sensors way, most of the electroactive region should be around 0 to 0.3, 0.4 volt region. Okay, in that region, in biomedical application, whatever the possible analytes are there, neurotransmitters, whatever the drugs are there, those are active in that region. But you can label some of the redox probes labs, and you can measure those potential in the far region. Means with amperometry you can apply the, so some label, redox label is there, you are modifying with the aptamer or antibody or with enzyme, but that label molecule can exhibit the signals around maybe minus 0.3, 0.4. Okay, so in that range you can choose the amperometric range of that one only. So in that, the possible way, the positive potential, whatever you're talking, the 0.3, 0.4, those are the major region it can available in that, and those will not participate in the reactions. In that way, it's also you can choose one that way. And other way that you can use certain type of the coating layers. So in that coating layer can attract only certain type of elements, and certain type of elements can reflect. Or you can use as a polymers, and that can allow certain type of molecules, and that cannot allow certain type of molecules. In that way, we can achieve over the sensor, at least for the real matrix analysis.
I have another question from the audience. So
Okay.
what was the typical response time when you used these type of sensors, wearable sensors?
Okay. So the response time is whatever we are measuring, it's continuously working actually. Means we cannot say, means the response time we will get in 30, 40 seconds in amperometry. So if you induce the 30 seconds time, that you will get the response, output value you can get. But we can make our own device. So we don't want in every minute reading. Suppose you can make it as that maybe every 5 minutes you will get the reading, or every 10 minutes you will get the reading. Like that, you can mold yourself and while making the device. So that is enough. So sometimes you don't want every time, like how in the watch we are getting the heart rate every time. There is no not, but every half an hour you are getting your glucose level, that is fine. So that way also. Typically response time in the minutes only, below minutes only. Either you can use the pulse oximetry technique or amplitude technique.
Some are interested on listening about agricultural applications. I think
Oh,
you would be
agriculture.
half
Okay,
of
okay.
that.
So in agricultural expansion, maybe I'm explaining the next part. So in agricultural expansion also, same thing. Whatever we are talking in biomedical here, the response time would be the same. Suppose you are using the potentiometric sensor. So maybe initially you need to calibrate sensor, you need to pre-incubate sensor. So maybe that steps will take long time. Pre-incubation steps, maybe 20 minutes, 15 minutes. Whenever the sensor ready, whenever you enter into the field, whenever you keep it in the soil, whenever you keep it in the water, whenever you keep it on the top of the leaf, top of the stems. So that time the response would start in the seconds only, 30, 40 seconds only the response would start. And it will keep on going. It will keep on going.
I will be looking forward to the next
Okay,
half
sure.
of.
Okay, next talk. Okay, next session.
We'll take the remaining questions at the end.
Okay, sure, definitely.
Yeah.
Then I will carry
Thank
on
you.
the next session. So till now, I talk about the biomedical sensor that is majorly a microneedle sensor. So one more the biomedical application that should be the sweat-based sensor here. So this is the ionotophotic based sweat-based sensor. So here, so what we are in this work, what we did means, so we take the one plain adhesive paper and we printed our sensor on the dirt. So by using the silver traces, carbon traces, we printed the sensors. So apart from the sensor working area, we also printed one cathode area and anode area. Okay. So here the cathode and anode regions, both. So we are applying the little mild amount of the current to the body. So that is typically in the milliampere, 0.1 to 0.2 milliampere. It's light current. So you will feel as a little jerk. So by applying the 10 minutes, maybe 5-10 minutes, if you apply that one, so what will happen under the skin? Whatever the ions can segregate to the anode and cathode, depends upon the charge mobility. Okay. And in that, so then you are using in anode pot as one specific pilocarpine gel you are using. So whenever it's aggregated, so some part the glucose will reach into the some electrode part, right? So in that part, the sweat can absorb by the pilocarpine gel. Means on the top of the surface of the electrode, you are producing the sweat, good amount of the sweat. So this we are calling as a uh, active, means the passivation means like, not you are not doing the exercise, you are by using the external source you are producing this sweat. So then after producing the sweat, so these the electrodes here we are modified with the enzyme, this ascorbic acid enzyme. So in this work we targeted on the detection of the vitamin C. Okay, for that we modified the ascorbic acid enzyme here. So then the response we mounted with the square wave voltammetry and amperometry techniques. So in this work, initially, we use the different type of the conductive material layers. So those will contain the different type of nanoparticles, platinum, palladium, rhodium. So we tested initially, we optimized everything. So rhodium is giving the good catalytic properties and good conductive properties. And on top of that, we modified the enzyme. So, and we mounted the vitamin C levels with the square wave voltammetry technique and amperometry technique. So and typically, so we tested initially with the buffer, and we tested and we calibrated the sensor is working very well. So later, what we done, like we the four subject people. So then we have took the vitamin C pills inside, and we apply the minute amount of the current to the body, and we produce the sweat on top of the skin. and we added our sensor patch on the top of that one, and we tested the vitamin C level. So you can see that initially it's increasing the levels. So the current value is increasing means is increasing the levels, and later part is decreasing. Means its consumption is there. Vitamins are consuming inside the body. So like that, we tested with the different people. So this with the intake of the vitamin C. So the without vitamin C intake, you can see. So there is no increment in the red values. You can see almost with the matching with the bare lines. So there is no response of the vitamin C output readings. So by that way, we tested in the tears, we tested in the saliva, and we also tested in the sweat. So like that way, we can focus on the alternative, we can focus on the other vitamins, other ions also we can focus by using the same type of ionophoric base, sweat-based sensors. And moreover, I said initially, this sweat-based sensor can also be useful for the generate the energy also. Whatever I'm saying here, after sweat generation, the sweat is there, right? So in that sweat, you can cleave the molecules inside that and you can produce the energy. Okay, so energy harvesting is also possible with the sweat. And that is a biomedical application. Now I'm entering into the one is the security applications here, we can call. Okay. So in this one, so first we develop the textile place solid state fluoride sensor. So solid state fluoride sensor, ISC sensor. Why we are calling solid state here means whenever we see in our laboratory, we have a pH sensor. So most of the labs we have pH sensor, right? So pH sensor, ammonia sensor. So those look like a rod, and inside they have a two reference cell. Inside silver chloride is there, and outside glass membrane is there. With respect to the inside solution of the pH and glass membrane pH, they are measuring the pH value and output concentration, and that indicating the pH value. So, but those we are calling as a liquid junction based techniques. But here we are using the all solids. Means the for that liquid junction inside is must. Always you want to fill the K cell inside the reference cell. Okay. So we want to transfer those kind of technology into the miniaturize on top of the strip sensor or textile sensor here. So here typically we are taking the reference electrode here, and reference electrode we are using this some kind of cocktail solutions. So those cocktail solution can store the ions for certain times, certain hours and certain days. Okay. And the working electrode, we are coating initially with the some of the conducting materials. So whether you can use the 2D nanomaterials or CNT, graphene, any kind of materials you can use that to increase the conductive properties of surface. And later we use the ion selective membrane. Okay. So those ion selective membranes are the this is one type of research. So the organometallic chemists are developing the ion selective membrane for specifically to detect the different kind of ions, fluorine, hydrogen, sodium. Okay. So here we use the tin-based ionophore we use. So those exactly matching with the fluoride cavities. So whenever it is the fluoride reaching to in the surface of the working electrode, in the reference electrode already before working condition, what we are doing here, we are incubating with the fluoride solution. Okay, we are incubating the surface with the fluoride solution. So then it can exhibit certain potential value. But when you change it, that one, when you expose to the fresh fluoride solution value, you can observe in the change in the potential value. Okay. So the change in the potential value corresponding to the your fluoride concentrations. While you changing the concentration of fluoride, you will get the various other types of output of the potential value. So like that, we developed the first fluoride sense ion selective electrode sensor that we developed on the strip sensor and that we developed on the textile sensor. So then the major part of application is to detect the nerve agents here. So nerve agents here, G-type nerve agents. Okay. So and these fluoride sensor getting the good LOD, good limit of detection. So later we coupled that fluoride sensor with the enzymes here. So the enzymes is the organophosphate hydrolase enzyme or organophosphate acid hydrolase enzyme. Okay. So those enzymes can specifically cleave these the G-type nerve agent molecules. Okay. So for that mimicking the nerve agent molecules, we take as a similar type of organic compounds, because to access the nerve agents, it's not easy. It's not easy to do in the laboratory conditions. So we mimic the same type of structures. We use the deoxyfluorophosphate structures here. So the PF bonds. So these PF bonds can cleave by this enzyme molecule, and we release the fluoride ions. So those fluoride ions can detect with the fluoride IC sensor. Okay, so indirectly, by detecting the fluoride concentration, we are detecting the initial compound of the G-type nerve agents. Okay, like that we proposed the sensor, and we tested not only the liquid phase, we also tested in the uh, vapor phase. We took the we vaporized the solution on top of the textile surface, and we tested the sensor. So in that conditions also, the sensor can capable to detect the DFP and up to 50 micromolar concentrations. And similar way, so we further develop the same sensor by using the MAPs, because we realized that the enzymatic sensor is not working above 30 temperature, because 30, 35, 40, when you increase the temperatures, enzyme activity is decreasing. Enzyme is not acting very well. So that's why to alterate that one, so we use the biomimetic MAP molecules. So we tested the various kind of molecules, MAP202, EO66 amine, and MOF808 amines, and all are zirconium-based MOFs. So these zirconium-based MOFs have a good catalytic activity towards the PF bond cleavage. Okay. So, and these PF bond cleavage, the output we are getting the HF products, means fluoride is we are getting. So then instead of in the previous work, we use the enzyme. So we replace the enzyme with the MOF here, and we tested the sensor, and we detect the fluoride ions by using the potentiometric techniques, and the fluoride ion concentration respect to the G-type nerve agents. Okay, and these we demonstrated with the same thing. We took the nebulizer, we take the organic point of the G-type nerve agents, whatever mimicking we are taking, and we spray towards the sensors on surface, and we remotely tested with the with our mobile phone. Okay, and we got a good results in the micromolar range. And one more thing is that we tested the sensor in the various conditions, temperature in 25, 30, 35, 40, 50, up to 50 we tested. So the maps are little stable structures, so it's not the activity is not disturbed. We are getting the similar response in the room conditions and a very high temperature elevated conditions. But in the this the bottom part is the enzyme part. So in the room condition we are getting good way, but in the high temperature condition the activity is losing. Okay, so that way we develop the green mock based textile based sensor for detection of the nerve agents. This is one of the application of the security applications. A similar way for in industrial purpose, if you want to detect any toxic molecules or volatile organic compound molecules, you can focus on the sensors and the textile based sensors, and you can work out on that. But one thing is that here we need to take care about the flexibility, stretchability. You want to use the good type of things, because whatever the normal traditional sensor, it's not possible to use in these conditions in real, because in mechanical stress, those connections will be lost, right? So you need to keep it in mind. You need to work out on that area also. So then coming to the future work possibilities of these electrochemical, wearable electrochemical sensors. So majorly in 4 domains it can work. One is the agricultural, food environment, and biomedical, and security, and along with that energy harvesting also, that we are calling as a biofuel cells and bio supercapacitor. Okay. So how we can use these techniques in agriculture and food and environmental field? In agriculture, as I said, as India is a developing nation, the usage of the herbicides, pesticides is very high. So there is no control in the monitoring. So if you imagine that you have a sensor that can detect the phosphate levels or pesticides levels, herbicide levels during the field, agriculture field. So by that way, you can reduce the content of that. Okay. So by using the potentiometric sensors, solid state potentiometric sensors, you can develop the potassium based sensor, carbonate sensor, pH sensor, moisture sensors you can develop. And at the same time, you can also develop the pesticide detection, herbicide detections by using the wearable sensors. Means you can attach the sensor to the leaf or sensors to the stem, and whenever you're spraying the herbicides, you can find out what the concentration is there. So whether you want to add more herbicides or not, more pesticides or not, you can control. And in the water levels also, how whether by detecting the moisture levels, whether you want to give the more water to the agricultural fields or not, you can test it. And the pH also you can test, soil pH also you can test. And similar way, you can test the mycotoxins and pesticides. This is one of the good area in agriculture and environmental field. You can implement these textile-based sensors, strip-based sensors, microneedle-based sensors, and plant that we are calling as a plant wearable electrochemical sensors. And one more application is the biomedical applications. So as we know, in biomedical application means major thing is two things: biomarkers detections and therapeutic drug detections. For chronic disease management, the drug intake is very high, right? So till now, the major research is focusing on only the diabetic management. So whatever the devices available, only in the diabetic field only. But there is a plenty of opportunity to work out on the other chronic diseases, neurotransmitter diseases, and cancer, cardiac, renal. So, and all of these wearable sensing platforms can extend to the other chronic diseases for the detection of the biomarkers and for the detection of the therapeutic drugs. And the same time, infection drug diagnosis also, viral detections, pathogen detections also. Suppose you imagine you have a glove, you have a sensors on the top of that one. So you want to check the virus levels on these in office or in the public places, you want to test whether virus is there or not. So you can touch it and at least you can get the preliminary information of the pathogen information, virus information. That is also possible with the glove-based sensors. A similar way, the security applications, security, as I said, the drug market, illicit drug market and forensic drugs market is very high in India. So nowadays we are catching the plenty of illicit drugs, methamphetamine, cocaine, morphine cages in the port, especially in Punjab, Kerala, and Gujarat, in the portal area, we're finding very high. So these wearable glove-based sensors, imagine you have a glove sensor. So these gloves can modify with the uh, chemical recognition elements or optimal recognition elements or MIP-based recognition elements. So those you can touch the liquid part or maybe the solid part, and you can get the preliminary information of the whether they have it's a normal powder, they have any cocaine or morphine. That way you can detect. Okay. And as I said, but chemical warfare agents. So just I focus on only the G-type nerve agents. So there is a plenty of nerve agents are there. So to detect that one, you can develop the sensor, textile-based sensors you can develop. So majorly, I'm saying the biomedical sensors, the microneedles, and agricultural security sensors, the globe-based sensors. And one more thing is that, as I said, the biofuel says bioenergy. So to the sweat and urine and saliva and tears, whatever the biofuels, in that several components are there, biological components. Those you can cleave with the bioiners and biocathodes, and you can generate the energy from that source. So that is also one of the important area, bioenergy area, biofuels is something. One thing I stress here, so when you want to achieve the final product and final prototype design, you want to develop, so you want to do the collaborative work here. Here I can say that any one of the chemists, any one of the physicists cannot do alone here. So in this type of work, we need a collaboration works. So means we need expertise in the fabrications, printing technologies, and we need moreover the miniaturized electric devices we need, because we cannot detect with the bulk instrumentation techniques laboratory. When you want to go to field, you have a small miniature techniques, you want to connect the sensor with that one and you want to test it. So to achieve the prototype design, to achieve the products, you want to collaboratively work with the different fields. Majorly, I can say that chemist, physicist, and along with that one, electric people, and moreover, nano engineering, nanotechnology people all need to combine and can work in this field. Just for example, I can say the agricultural field, how we can focus, as I said, you can make a sensor, strip-based sensor, paper-based sensors, or tattoo-based sensors, and modify them with the unselective membranes or any other optimal assays or enzymatic assays, any assays can make it. And you can focus on detection of the pH, moisture, stress markers, potassium, carbonate, phosphate, pesticides to focus. And the detection only not on the leaves, you can focus on the soil and water and all plant physiology. So you can focus. So there is a plenty of opportunity in this field, wearable plant sensors. And nowadays you can combine with the AI technology, you can develop your own apps and sensors, and remotely you can monitor also. And this is one of the current project we are doing here in our lab. So we are focusing on the detection of the methamphetamine and fentanyl illicit drug molecules with the help of the MIP and abdominal based sensing technology. So we are focusing on the detection of the powder molecule detections, illicit drug detections. Thank you. I would like to acknowledge all of my previous institutes where I worked is San Diego and Johns Hopkins University and my current institute, IIT Palakkad. And moreover, I would like to thank SEB India for the Ramanson Fellowship. And this is my research group. What is sure? Okay. Any questions from the audience or we can discuss our
Detailed presentation. It was very nice. I have a couple of questions from my side first. During the chloride and flow sensor, you mentioned that for checking the real-time applicability, you vaporized the sample. How did you do that?
So means just to mimic the vaporization, what we use commercially we have nebulizers, right? Commercial nebulizers we have to for children if you want to add any type of they have cold conditions and you have a small nebulizers are there. Okay, so the nebulizers what we can do when you add in one top of the liquid and little it can heat it and it can place as a vapors. Okay, so like that the vapors we force on the sensor circle. So we took we mimic the glove box and we have one hole. From that, by using the nebulizer, we sprayed that one because we don't want to expose to the that molecule because little we toxic molecules. So by that way, we remotely connected means the sensors is on the textile along with the potential is also inside only. So whenever we vaporize, we waited for some time and we mounted the readings in the our mobile phone. Remotely monitor.
Second question I have is, you mentioned about zirconium marks.
Okay.
Do you prepare the lab or do you purchase from commercial suppliers?
No. We prepared actually. During my UCSC stay, that work is did by the collaborative work from chemistry. We are from management department. So we prepared some of the MOF, MAP 202, but some of the MOFs we prepared in the chemistry laboratory with the help of the professor Seth Cohen. So he's also author in that JAX paper. We took the help from that group. Okay, because some of the characteristic techniques not available in that our group. So we have taken the help from them because he's expert in the MOFs field. So our his work is on the development of the MOFs, how we can develop effectively synthesize the MOFs to get the more activity, more catalytic activity towards the phosphates, nerve agents. So we took the help from that group.
Extension to the same question.
Okay.
If you find commercial suppliers, that would be interesting. So that any.
I think I never thought about that one, but I hear that some of them are supplying from the Taiwan and Korea. They're supplying, but we don't think so. We can able to synthesize in our lab because that time our laboratory is majorly focused on biomedical things there. So we don't have good facilities to focus on the material synthesis there. So that's why we took the help from the other group. But now I'm also planning to work on that field. But we can possible. In India, a lot of people are working in the MABs, good universities, all IITs.
Let's take one audience question. The first one is, you explained about nerve agents, G-type nerve agents.
Yes.
Then what would be the simple difference between G-type nerve agents or other nerve agents versus, let's consider island. So how do we distinguish?
So it depends upon the PA bond category, means P bond category. P phosphate inside that nerve agents, how it is, what type of ions is connecting with that. So those are category with the V, G, B type, different type of nerve agents. So means if it is attached with the chlorine, we can call as one type. If it is attached with the OH, we can call as one type. If it is attached with the fluorine, we can call as one type. If it attached with the CN, we can call as a one type. So those categorization. So that I know from the literature only. I'm not very expert in that, but so I know that the fluoride connecting phosphate nerve agents we are calling as a G type nerve agents. Fluoride cyanide connecting nerve agents.
How do how can we distinguish that with the effluents from oil and gas industry? That's the question from audience.
Oh,
Let's take
how
two
we can
examples.
okay. How we can distinguish between the normal nerve agents and the oil and that one, right? Okay.
Yeah.
So in most of the oils means it's a combination of the it's a kind of polymer kind of solutions are there. I don't think so. If the oils they have the fluoride, chlorine,
Correct.
and cyanide ions. So the sensor whatever we are developing that basically working principle is that specifically detecting the fluoride ions. Okay, so even in the presence of the other nerve agents also, because the main whenever you want to develop sensor, the main interference will come from the nearby structural samples. Means suppose nerve agents, the similar type nerve agents, whichever they have the other group, if you even if you detect in presence of that one also, so then it can possibly detect with the others also, because others there is no interference that will not interact with the, maybe that can act as a falling, maybe that can inhibit the sensing performance, that we want to test it actually, because whatever we tested with the simple buffers and in the liquid solutions only, we haven't tested in the real conditions, and along with the other things we haven't tested. Okay. That we have to do.
Interesting. Where do you like? For example, you work on serotonin detection
Okay.
or other
Okay.
detection,
Okay.
and also wearable sensors. They want to
Mm.
know where do you apply that patch or wearable platform to detect
Okay.
serotonin in body. Can you comment on
Okay,
that?
so good question. Comment, sure. Okay. So the thing is that the availability of the neurotransmitters majorly made it's related to the neuro, right? Means during the, but head part would be difficult. We cannot pierce into the easily into this one. The best part is to contain the more neurotransmitter in this part, but I think you can connect on the normal ISF anywhere also. You can connect. Means with arm also you can connect. Still in ISF, toward the body part, the concentration is there. But the more abundant part is the nervous near the nervous system. So the best part should be here. But thing is that we want to develop the needles. Those can pierce into that, and we cannot harm to the head also. But we can wear in the
And
normal arm also. Arm is the good place, and that would be the good part.
another question was related to biomarkers detection.
Okay.
You were saying like we can extend to any type of chronic diseases. So the question was, can we detect prostate specific antigen (PSA) by using these type
Yes,
of
yes. Yes, obviously. The research is going on, especially for cancer biomarkers detections. Research is going on. But major thing is that wearable platforms, they are not focusing. The first aim is that in that point of care sensing, they need to achieve. So whatever we
yeah.
are talking the wearables, someone already explored more in the point of care sensing, and they already achieved. So the same sensing strategies and technologies, we are trying to improve that one and to go with the continuous monitoring. But that is a good field. We can aim at that one, because suppose imagine that for breast cancer, you have a device with a nipple. You can wear one thing on that, and maybe with a bra. So inside the bra, you have a sensor. Okay. So that can also can monitor the your biomarkers, breast biomarkers levels, and that can keep the initial readings and thoughts. Okay. And already if you are diagnosed and what is the status. So in that field is also people are started working, and in future it will go on. That is also in cancer field is also is there, and especially for cancer biomarkers in the saliva also. So for throat cancer, so the probability of the biomarkers is more in the saliva. So you have a, suppose you have a mouth guard. Otherwise, teeth, you can have a sensor on teeth, and in saliva. So whatever the biomarkers available in the saliva, so that also you can detect. So in that region is also people are focusing. So
The next question.
okay.
Let's take Apple Watch or any other smartwatches. Can we integrate this type of wearable sensor platform which can detect biomolecules using like glucose or lactate sensing, etc. Can we
Yes,
integrate that? Would be the challenges in
yes,
that
yes,
integration.
yes, yes. So now almost the Google and Apple and Tesla. So they invest almost means $1 to $1 billion in this market. Okay, and the whatever the continuous glucose monitoring, they are aiming, especially for the Apple Watches, they are aiming to integrate with Apple Watch to know the glucose levels. So maybe currently they are focusing on the biophysical method. I mean, in biophysical way, they're focusing, and even electrochemical way also they're focusing. They're working on this field. So near the in San Diego cluster, near the Los Angeles clusters. So the Dexcom, they're focusing on the device, this one, but Apple and Google is also investing the money. Maybe in the future, in the nearby future, some devices would come. The Apple also can keep the glucose rate levels along with
In
this
that
technology.
in that conversation, okay, I'm interested to know.
Okay,
It's my personal question. So
okay,
let's take COVID and flu, all this type of detection. We are right now depending on the PCR technique or
yes,
any other conventional technique, gold standard technique.
okay.
Do you think this sensor technology can replace them? If, how long it can take? Like when can we expect that? Do you have any idea on that?
So my comment and my view on this one, I can strongly say that it cannot totally replace them. Uh, it can act as a supporting role for that one, because the main thing is that accessibility. So why we are focusing on this one is main thing is that accessible to the patients and personalized device things. Okay. So I don't think so it will replace, but in terms of this, some disease it can replace. Suppose if you are maybe the diabetic management nowadays, no one is going for the testing to the laboratory. So even they are doing with the glucometer, nowadays they are doing with the continuous glucose device things only. Okay, but I don't think so that will totally replace with all of the sections, because why the glucose sensor like heated more means why not other sensor means. The major thing is that concentration levels of the glucose is very high. And it is in millimolar, means the abundant concentration is very high. So that's why they easily develop the sensors in the point of care things and the continuous glucose monitoring things. But whenever we talk about these, the cancer biomarkers and cardiac biomarkers, whatever we're talking, so those biomarkers levels are very like minute levels, sensitive levels. So those we can get the preliminary idea, but especially uh, then it will give support to the hospital. Means it will give an alert to them, so you can go and check the reports, and you can meet the doctor. Okay, as like.
The answer is
Uh,
simple. Whether
but in
we
agricultural field, I can say that in agricultural field and security field, it's possible, because in that case the concentration abundant is more. There is no sensitivity part is there. But especially in the biomedical part, that too in the cancer, cardiac, renal disease management. So the sensitivity to the plays major problem, because the biomarkers levels are very less there. But in coming to the agriculture security application, there is no concentration problem. There means the abundance is very high. So in that case, it's possible. So you can you don't need to depend upon others. You can make the sensors. You can slowly like mainly depends upon the electrochemical sensors also. With pH, moisture, potassium, phosphate, everything if you develop the sensors.
take another question here?
Okay.
People are interested, our audience are interested to see whether have you considered any climate changes, rainfall seasons. For example, you suggested and you worked on agricultural products. Did
Yes,
you
yes,
check
yes.
application in during rainy season versus summer season? What was
No, no.
what influence?
Uh, so currently we
Comment.
have an currently we have an agri-tech in IIT Palakkad. We have an agriculture center. So at the moment we are focusing on the soil sensor. We are focusing. So the different conditions means we are taking the different part of the soils, and we are testing the carbonates and pH of the solution. That one. So obviously, so we are not working on the whatever I gave in the last slide, two slides, the possibilities I gave. So at the moment, we are focusing on the pH and moisture only. Slowly, we can focus to the pesticides, herbicides, and carbonates, all other ions also. But obviously, you want to check all the possibilities, different seasons. You have to focus different part of the soils. You have to take it, different
Collaborate with any,
temperature conditions. Huh?
do you collaborate with any other already established groups who are working on this type of agriculture applications?
No, here we are interdisciplinary on electrical engineering people, and we are working, and we are collaborating with the one University of Texas, Dallas. One is she's also Indian. She's from Shalini Prasad. She's more oriented working on this agriculture oriented sensors. So recently she developed the device for the uh, detection of the carbon levels in the soil and the pH levels in the soil. Okay. Moreover, so we are closely following her and we are taking the advice from her. Okay. Thing is that one of my master students also going for PhD to under her. So he inspired her work and he's going to join in the next session. And we will try to collaborate with them in future.
Yeah, thanks a lot for that extensive and nice presentation. And thanks for sharing your versatile erudition on this field of research.
So maybe one question from my side. So since you are working as an industry, so all of the academic research is majorly focusing on the up to the prototype designs. Like whatever we focus, the prototype only. So the how far like the prototype design means until we are working with the small conditions only. If you want to deliver into the product, what are the major challenges we can face? How we can do?
So first of all, the sensing mechanism, we should extensively establish that whether it works at any under any conditions. Let's take temperature. Within India, let's consider down south, the climate, temperature, humidity, that's different compared to towards north, near Himalayan region. Within India, if you want to develop a sensor, it should work the same, right?
Yes.
But the temperature and humidity, this will influence a lot. So we need to develop the recognition matrix such that it works in any extreme conditions. If not, we need to address that. What would be the ideal conditions we need to use? Second comes like, for example, in the laboratory scale, we work with maybe maximum of milliliters of the milligrams. But if you want to extend it to the commercialization, the problem is reproducibility from the lab scale to bulk scale. So the same exact recognition matrix in 1 gram or 1 gram versus kilograms, the dimensions and entire
Or maybe
picture
while preparing the matrices, so it can change. Okay, small scale and big scale.
influences. So
Yes, it
we need
influences.
the sensor performance using the calibration plot, whether we are
Okay.
getting quickly or not. Most of the times in laboratory, if we get 3 times or 4 times, we will leave that. But we need to get 100% on each sensor when we go for commercialization. For example, 10 out of in lab, at least if we test 10 sensors, maybe 8 or 6, sometimes 5 also works, and we discard the remaining. In the industrial case, we can't discard, we can't do that. So we need to address that.
Yes,
That will be
we need to achieve at least 99% we need to achieve.
That's the challenge. We need to produce the sensor in a bulk scale with highest reproducibility. That's one key challenge. Because it's not just about the recognition matrix. It comes with a lot of other parameters. For example, you are fabricating in a commercial plant. So lots of other issues come into picture, which we don't even imagine. So that's really interesting.
Yes. I imagine because suppose you want to and plan means the mechanical stretchability, bendability, you want to try with the different inks and it should work in the extreme conditions. Whenever you twist them, even though the matrix should not leave and connection should not leave. Okay, that should be we should take care, especially in agriculture and security. So I agree, and then even in the biomedical also, because just now I explained in the initial presentation, we tested only 4%, 5%. That too in our lab people only. But if you want to go with that one, means we want to test with the more people, more population. As we know, as how the vaccines has been tested with the different age people, different kind of people, different gender people, different continent people. So we need to consider all, whatever the focus market. So you need to consider all the things. So the best example, as we said in India. So if you want to commercialize in India, you want to consider the all extreme conditions, not east, not south, all conditions. So in different conditions and different people, we need to test it. This is that we need to do. Good.
Yeah.
Okay. So I think
Yeah.
we can invite so Professor Rapa, Mr. and Sushil.
Thank you very much. This was an absolutely fascinating webinar. Yugender Goud Kotagiri from IIT Palakkad and Koteshwar Reddy from Identify Sensors Biologics Corp USA. Thank you so much to both of you for having shared so much with us today. Especially because of the wide range of applications that we touched on and the possibilities that it opens up. This is a very, very fascinating area. And I think a lot of people have been commenting and they're saying that, they're very, very, it's absolutely fascinating and interesting to know that there is so much work happening in this space. Once again, to our regular webinar viewers would recognize this. This is again a multidisciplinary area in terms of the applications, absolutely wide-ranging set of possibilities, and even in terms of actually building these sensors and making them work, also very, very multidisciplinary. When you talked about wearables and said that even plants can wear them, I found that really fascinating. So today we saw several applications of these wearable sensors. We saw biomedical. We looked at therapeutic drug monitoring and delivery. We looked at infectious disease diagnosis. In the area of security, we looked at illicit drugs, we looked at forensics, chemical warfare agents detection, and in the industrial area, toxic or flammable molecules, detecting them. In the area of agriculture, soil health, herbicides, all of that, adulterants like heavy metals, for example. A very, very interesting new application, I thought, was the biofuel cells generating energy from the human body. And these are based on many different materials, very different combinations. What was again interesting was the fact that there is a wide range of possibilities and there is no single mechanism or single method for it, but many, many possibilities exist. We saw in detail the principles behind the design of electrochemical sensors, chemical, antibody, enzymatic, and so on and so forth. And again, interestingly, the fact that you can combine these methodologies to create an effective sensor. Key challenges and characteristics, and there was a whole page full of them, but some of them are usability, real-time detection, affordability, sensitivity, and so on. And then we saw a few examples of how some of these can be addressed. We went deep into the on-body sensors and the different sensing platforms such as microneedle, glove, textiles, the socks, for example, tattoos, and how they are developed. And thank you for sharing all of that information, Yugender. Some of these, you did say, are in the development stage, whereas some are available already for use. Microneedles detection and delivery of drugs, and you took the specific example of degenerative diseases and talked about levodopa for Parkinson's. The process of fabrication of the microneedle array, again, so many different options that are possible. Tattoo sensors, what they are, how they work, all of that. And then we ended with a very interesting discussion around what is required to commercialize what is happening in the lab, the developments that are taking place, and the kind of challenges that are there. We all look forward to seeing a lot of these applications move into the commercial space. And we hope that many of people who are listening in today are going to be inspired to take up some of this work and move it forward. And many thanks to both of you for this wonderful webinar. Koteshwar, particularly appreciate your waking up in the wee hours of the morning to join us. And to our audience, thank you very much. Some of your questions were so interesting, and they brought the webinar to life in a lovely way because of your questions and your interaction. We look forward to welcoming you to our next webinar. The recording of this webinar is going to be up on our YouTube channel. Please look for the IITACB YouTube channel. It'll be up come Monday or Tuesday. And please feel free to share this with anyone who might find this of interest. So thank you very much again, and good evening.
Maybe can I take 1 minute? So
Yes, please.
I would like to thank the Dr. Sushila and Dr. Abha Mishra, so providing this opportunity and providing this platform to share my research experience to these students. So thank you very much. Many thanks.
Thank you very much. Goodbye.
Let me also say very much, Dr. Sushila and Dr. Abha. Thanks for your time and for giving me this opportunity. It was a nice talk. Thank you.
You're absolutely welcome. Bye.
Yeah.
All right.